Thanks for that splendid introduction, John. He's very modest. I mean, I won't take too much time to say this, but he's really transformed the field of diagnostics of fusion oncogene-driven cancers by developing a technology platform that is broadly, universally used, and is in fact one of the reasons why RNA is very important in these trials that you do, because RNA is the best way to detect fusions, and he's the one who figured that all out. But okay, so let's see if I can figure out how to get this to work. Okay, good. So the tumor that I focus on, as John mentioned, is called NUT Carcinoma. It has changed from NUT midline to NUT Carcinoma, and I'll explain that in a second. So the tumor is, this is a rare cancer of patients of all ages that is actually a subtype of squamous cell carcinoma, which is very unusual for a squamous cell cancer to be driven by a fusion oncoprotein. And it is defined by rearrangements of the NUT-M1 gene, which is actually a testis-specific protein or gene. So it is defined by those, meaning that every tumor has to have a rearrangement of NUT. But they do have a variety of fusion partners, the most common being BRD4, this gene right here. So 75 to 80% of cases actually have a BRD4 NUT fusion. But like Ewing type tumors, it has a bunch of other alternative fusions, including BRD3, which is highly related to BRD4, as well as NSD3, and some really small, rare cohort of ZNF NUT fusions. Now, these are all related in the sense that they all function normally in a BRD4 transcriptional complex. So they actually are interchangeable as fusions to NUT, and they pretty much do the same thing. It means all these tumors are sensitive to inhibition of BRD4 or NUT. A few words about the sort of the structure of the protein. I am going to get into some science, so I apologize for those of you who might glaze over with some of that. I don't know how to make it more interesting. I'll do the best I can. Okay. So the NUT gene is an unstructured protein. It has two different domains here that interact with other proteins. And the BRD4 gene, though, protein is much more structured. It has two chromatin readers. These are bromodomains that bind to acetylated chromatin, which is associated with active transcription. So that reads actively transcribed DNA. And those with the two bromodomains. And it has what they call an ET domain, which is an extra-terminal domain which binds to other it's a protein-protein interaction domain. All of this stuff is this business end of the protein is fused to NUT in every single case. And so moving on, I have a timer here, this good? 17 minutes left of my talk. So we did call this NUT midline originally because the vast majority of tumors arose along the midline in people. And we actually initially thought this might be a germ cell related tumor, possibly. It turns out it's not. It is a squamous cell cancer. But the most common site of involvement in patients is the thorax. That's 50% of patients. And then another 40%, it affects the head and neck. Just the way squamous cell cancer, that's basically the same distribution of squamous cell cancer. This is tragically a tumor of mostly adolescents and young adults. So it's what we call an AYA gap tumor, which would also include germ cell tumors as well as Ewing sarcoma that some of you may be familiar with. Also probably the desmoplastic small round blue cell tumor is also probably an AYA tumor. Dr. Banks could correct me on that if I'm wrong. But the age distribution, patients of all ages are affected. In fact, there's a huge testing bias for youngsters. So we don't even know if this is really the true distribution of ages. This is very likely there's a lot more older folks who are getting this cancer. They're just not being tested for it. NUT carcinoma is unquestionably a rare tumor. It's also unquestionably underdiagnosed, and that's probably the case for a lot of rare tumors, is that they're underdiagnosed. If you look at the distribution of testing, this is a cohort of 412 patients, you can see that there's a lopsided increased number of cases in the Northeast of the United States, which is basically where we do the testing. And also in Europe, where they also do the testing. But if you look at, doesn't correlate with population. Okay, so we have not that many patients in Africa or India or hardly any in the Soviet Union. Purple is high density population. It's more correlated with developed nation and also with testing, which means that it's being vastly underdiagnosed. We estimate about 1,000 to 1,400 cases per year, which by comparison with another rare tumor, Ewing, it's about four times more prevalent than Ewing sarcoma, probably because the age distribution is broader. It is a tragic tumor. It is one of the most aggressive solid tumors known to man. It is second only to anaplastic thyroid cancer, which was earlier mentioned, and has a median survival of 6.5 months. So this is a really devastating disease. The oncologists describe it as a Burkitt lymphoma without a treatment. I'm going to get into some of the sort of the mechanisms for how this tumor grows. So BRD4-NUT, the BRD4-NUT fusion, its main sort of overall function is to block differentiation of tumor cells, so that instead of differentiating and going through the normal cycle of maturation and then senescence, the way most healthy cells go through, these cells are perpetually young, and like Peter Pan. Except malignant. And so if you look at NUT carcinoma cells at baseline, they are very undifferentiated. You could not tell the difference between this and a leukemia or a lymphoma or a germ cell tumor. They all just look kind of non-specifically undifferentiated. If you knock down the BRD4-NUT oncogene using siRNAs or other means, they will differentiate very rapidly into squamous stratified epithelium that stops growing. You could literally peel a skin off the dish after you knock down the BRD4-NUT. I wish we could get RNA, siRNA into the body this way, but we can't. It does tell you that BRD4-NUT blocks differentiation, and then it's an excellent therapeutic target if we can actually target it. Okay. The other fact about BRD4-NUT is that it alone can drive tumor formation, invasion, and metastasis. And we know this now with genetically engineered mouse models of the disease, where we can induce the formation of the BRD4-NUT fusion, as we're showing right here. And it has incredible penetrance and very rapid growth. So what we have found in our mouse model, and there's another mouse model out there which shows essentially a similar finding, is that 100% of mice where we turn the BRD4-NUT on will get tumors, and 100% of them will die from those tumors very rapidly. Here's the tumor formation in blue and survivals in red. It looks a lot like the survival curve in humans. Okay. H&E doesn't look, as a pathologist, this is a little bit hard for me to see because it's way blown out here. But anyway, so we have also found that there are in situ lesions, meaning before the tumors become invasive, there's a pre-malignant lesion within the squamous epithelium of these mice that's created by the BRD4-NUT, indicating that these really arise from squamous epithelium. They're not coming from a germ cell or something like that. And also the tumors in these mice are poorly differentiated, just like the human tumors. So again, the point is the BRD4-NUT is a lone actor. It doesn't really require help from other mutations, like a lot of cancers do require that, not this fusion. And so getting to the normal function of BRD4 and NUT, the sort of physiologic function, BRD4, as I mentioned, is a chromatin reader. And so it will read acetylated histones, which are associated with actively transcribed genes. So it's attracted to genes that are being transcribed. And we'll go there. And it'll bind to the histones. The NUT, on the other hand, is a recruiter of the P300 histone acetyltransferase, which is a writer. It writes, this P300 actually creates the acetyl post-translational modification on histones that lead to increased transcription. So when you put the two together in the BRD4-NUT fusion, you now have a reader-writer, which basically supercharges gene transcription and activation through the combined reading activity of BRD4 with the recruiting activity of NUT. And what actually happens is the BRD4 NUT, in its ability to read open chromatin and then to recruit P300, is it creates its own acetyl marks, because it can now write, which then recruit more BRD4 NUT. And it has a feed-forward process, a snowballing effect that creates these gigantic super enhancers. These are essentially insulated regions of open chromatin that will just maintain transcription of the gene of interest. And in this case, in the case of NUT carcinoma, the gene that gets targeted every single time in every tumor that we've analyzed by BRD4 NUT is the MYC gene. MYC is a well-known proto-oncogene that drives growth of cells. So NUT carcinoma, in fact, is actually a MYC-driven tumor, like tumors like Burkitt lymphoma that some of you may have heard of. Very aggressive. MYC-driven tumors are highly aggressive. So you can actually see these, what we call megadomains, or gigantic super enhancers. You can actually see them by immunofluorescence. They form these nuclear condensates, and they are co-localized as expected with acetylated histones. So we have a collaborator of ours, the Egan Lab at Baylor, has actually looked at what's happening three-dimensionally on chromatin when BRD4 NUT is around. And it turns out it forms its own little subcompartment, three-dimensional subcompartment. When BRD4 NUT forms these megadomains start to interact with each other from long distances away. This doesn't normally happen. And they actually form a tertiary kind of three-dimensional chromatin complex that brings together genes from all over the genome, from different chromosomes, so interchromosomal and intrachromosomal interactions to form these. So when we look at these foci, you're actually looking at DNA from long distances coming together in like a group. It's like we're having a big party here. This is actually the data showing that there's a ton of interchromosomal interactions that are occurring as a result of these megadomains that are formed. And one of the important interactions that occurs is between chromosome 5, which has SOX2 and P63 on it, and MYC. So we're basically bringing megadomains together from the three major survival and growth transcription factors for NUT carcinoma, which are P63, SOX2, and MYC, together so they can reinforce each other's expression. So this is like just a very evil mechanism. It's super potent. This is all susceptible to inhibition with a BET inhibitor that I'll get into in a second. So BET inhibitors I don't want to say they were discovered here in Boston. They were actually discovered by Mitsubishi Tanabe in the 1990s, but they were repurposed, or rediscovered, I should say, in the 2010s by Jay Brader. And together we developed or tested the use of these BET inhibitors in NUT carcinoma. That ended up being the prototype cancer for studying this new type of inhibitor. So what they are acetylizing mimetics that bind the bromodomain of BRD4. BET inhibitors bind the BRD4 binding pocket in BRD4, and they competitively inhibit the binding of BRD4 to chromatin. What this does is it phenocopies the differentiation that we see when we knock down BRD4 NUT. So you would think, oh, this is the perfect drug for this cancer. And you can see that it's actually highly active in mice, prolonging survival and also causing tumor regression. The answer, unfortunately, is that BET inhibitors, for a variety of reasons, including the fact that BRD4 is expressed normally in every cell in the body, have a very, very narrow therapeutic window. So we can't cure. We have shown in multiple clinical trials that BET bromodomain inhibitors on their own cannot cure this disease. They can give you 1 to 3 months of progression-free survival in patients that respond, which is about 20 or 30% of patients. And so it doesn't, on its own, BET inhibitors are not a solution, but we do believe that combining something with a BET inhibitor, we do believe that this is a nice backbone and starting point for combinatorial therapies, because it is quite specific for this fusion. And it is active in other types of MYC-driven cancers, by the way. So we sought to look at other susceptibilities in this cancer by looking at the other side of the coin. We've been focused on gene activation and proto-oncogene activation, pro-growth activation by BRD4 NUT. What about the other side? What about silencing? You can't really have one without the other. You can't have gene activation without silencing of another. Very often, tumors like to suppress. I got 5 minutes left, so I'll make this. I'll have to move fast. Tumors will have to. They not only do they have to have proto-oncogenes, they also have to suppress tumor suppressor genes like p53, which is the most famous tumor suppressor genes. So we looked at chromatin silencers that might be involved with this process of suppressing tumor suppressor genes, and what stood out was EZH2. So EZH2 is highly expressed in NUT carcinoma. This is very likely because it's a MYC-high tumor. MYC actually increases expression of EZH2. It's not a mutationally driven expression of EZH2. And we find that even low doses of tazemetostat, which is an EZH2 inhibitor, will result in, again, a phenocopy of the differentiation process that we see when we interfere with BRD4 NUT. And also, they're quite sensitive growth-wise. The growth of NUT carcinoma cells is quite sensitive with tazemetostat treatment alone. We wanted to know what tumor suppressors might be involved. So we did a targeted CRISPR screen to look for what genes, tumor suppressors that might be knocked out by EZH2 could lead to rescue of cells from tazemetostat treatment. And we found that what stood out quite obviously, and we've repeated this many times, is CDKN2A, which is a well-known tumor suppressor that encodes P16 and P14, which arrests the cell cycle. And so you can see that the CDKN2A gene is covered with H3K27 trimethyl epigenetic mark, which is a silencing mark that is specific for EZH2. And when we treat with tazemetostat, that disappears 100%, and you see re-expression of CDKN2A. We've also found that if we overexpress P16, which is encoded by CDKN2A, the cells stop growing. And we have found that BET inhibition combined with EZH2 inhibition is highly synergistic in these tumor cells. You can see these are NUT carcinomas, 4 different NUT carcinoma cell lines treated with very low doses of either tazemetostat or the BET inhibitor. And you can see that the monotherapy with either agent alone does hardly anything because it's a very low dose. But when you combine the two, it gives you this profound differentiation occurring, which is true synergy. We found, in fact, I'm going to skip forward. So there's synergy between inhibition of the repressor of tumor suppressors plus the repressor of proto-oncogene activators. Skipping to the money is the fact that we find that there's in vivo synergy between EZH2 inhibition and BET bromodomain inhibition. We saw cures in 2 out of 3 of our models with the combination of EZH2 plus BET bromodomain inhibition. These mice are only treated for 3 weeks, and after that, they're let to see how durable the treatment is, and it's quite durable in these 2 models. This one, we also saw synergy, but not cures with the combination of these 2 treatments. Now, I'm going to spend the rest, well, I'll just tell you. So basically our model is that NUT carcinoma is driven on the one hand by BRD4 NUT in these red megadomains that drive expression of MYC through the mechanisms I described, and then driven on the other hand by tumor suppressor mutually exclusive domains that are populated by EZH2/polycomb repressor complex number 2 that repress the expression of important tumor suppressor genes, and they cooperate together to drive the growth of NUT carcinoma. I have 1 minute to explain why we're not treating patients with this combination, and that has to do with the fact that, well, it's very hard to get any company to agree to combine these 2 things. This is actually where the NCI comes in, where they have a program called the CTEP program that will sponsor a compound to lower the bar for a company to allow their drug to be investigated in an indication that they don't necessarily prioritize. We haven't gotten there yet because we haven't gotten buy-in from a company yet, but there are some representatives in this room who are from a company that does make a EZH2 inhibitor, so hopefully I can talk to them later. But I do want to say that it's amazing how hard it is to get a company to agree just to let you try out their compound in people, and it's a barrier I'd like to break because we're just sitting here watching patients not survive and not benefit from potentially treating with this treatment.